All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Apologies for being a little late. The topic today is, I think, the most important at this time in COVID um, pandemic. The topic is about the long COVID and the possible pathology and the treatment. So, what I'm going to do is today's topic, I'm going to split in two pieces. This piece that I'm going to talk about will only have the basic pathology, high level discussion, not a deep dive, so that it can be small, this lecture. Number one. Number two, the vascular injury as a result of that pathology and the diagnostic method that is missing in the labs at this time, and how you can ask the labs for certain diagnostic approaches. And number three, the treatment approach. And again, please remember, this is not an advice. This is a treatment approach that you can discuss with your doctor, or you can pick up the original link of the study and then discuss that with your doctor. So, this, I think, why is this important? It is important because we are going to be exposed to this virus one way or the other. And the prevalence of the long COVID is a lot. I'll show you some data, anywhere from 30% of the exposed to 50% of the exposed, and more in women than men. So, with this prevalence, the amount of chronic diseases that would occur afterwards is going to be a lot. Pandemic will be finished, and this issue would continue, and the whole world's population is at stake. So we have knowledge of the S1 in monocyte from Dr. Bruce Patterson's group. Here is another important study, and I want to once again look at this part briefly. My request to you is look at the summary. I specially created summary in text form so you can have the overall picture of this thing in one slide. And then if you can share this with others as well, that is fine. If not, that is fine as well. Let's start our discussion. So here are the references. This is drbeen.com. Then here is the study about the global prevalence of post-acute sequelae of COVID-19 or long COVID. Now, I'm sure that this contains two types of long COVIDs. One is the tissue damage because of acute COVID, and then the result of that damage, for example, diabetes or the respiratory issues, or somebody's lungs are so severely damaged that they are oxygen dependent and so on. And then there is long COVID, for example, the long COVID we've been talking about, the tinnitus and the brain fog and the difficulty in concentration, issues with sleeping, tiredness, muscular weakness, and all of those things. So here, if you look at this study, why am I showing you this? To say that there is a greater chance of anybody getting COVID to end up with the long COVID as well. So look at this. These researchers took 40 studies and then did meta-analysis on them. So here is the result: 0.43 or 43%, and then 0.57 in the hospitalized patients. And then they say that it is more in women than men. So I would let you look at this, but just imagine that there is a very high prevalence of the long COVID after the acute COVID. And I hope my audio is working. I hope so. It was not working a few minutes ago. All right, so here is the work. So this is by Professor Resia Pretorius of South Africa. I am going to try to invite her as well and hope that she would join us. This is a very important work that she has done and her team has done. And if you see, they have actually multiple studies that they've done. So for example, here, TEG, microclot, and platelet mapping by guiding early management of severe COVID-19 coagulopathies. So thromboelastography, and I'll explain that. This is another study by them. SARS-CoV-2 spike protein S1 induced fibrinogen resistant to fibrinolysis, implication for microclot formation. And it just continues. This is another study by them. This is another study. And these are various links about what are the clotting factors. I have done a detailed discussion in the past about clotting factors and how they are disrupted. So this discussion today is not about the clotting factors. I want to keep it very, very focused. So let's now look at the work that I did. So here we are at the Humanities Coffee Shop, and these are the gifts for humanity. We'll continue. This is the summary. The summary is this. So they have multiple studies, and I have just tried to boil down in one page something that can be useful for all of us. Number one, microclotting is an important pathology during acute and long COVID. Vascular damage is not only in the lungs, but it is in the remaining body as well. We know that. And then there are important areas of damage, for example, vascular system in the brain, and then vascular system in other areas. Then there are important inflammatory markers that cause muscle weakness and the tiredness. Then there are important antibodies, autoantibodies that are produced, which also contribute. This is why their research, I had been spending the whole day today reading many of their papers. They are very, very thorough in their work. And then they say at one point that the autoantibodies and this whole pathology that we are discovering may be useful for chronic diseases like myeloencephalitis and the chronic fatigue syndrome and so on. Then the other important thing is S1 part of the spike protein, they feel, is the underlying problem. This is the same thing that we are seeing with the S1 in the monocytes from Dr. Bruce Patterson's work as well. Then they think that the clotting type is amyloid clotting. So I'm going to explain what is amyloid clotting again at a high level, but just keep this in mind at this time that this is a special kind of clotting. It's not regular clotting. Now, when you go for labs, normally the labs will come back and say everything is clear. The bleeding times are clear, the clotting times are clear, the D dimers are okay, the CRPs are okay. And the reason they say is that the markers that can identify the problem are actually themselves trapped in the microclots. So for the lab equipment to pick them up is not easy or not possible. So because of this, they say that there should be a specific type of testing done, or mostly the scanning electromicroscopy or fluorescence technique of the blood system to understand the platelet, RBCs, and the microclots and the things that are stuck in them. So perform TEG scans. And then they have said in their paper that the solution is antiplatelets and anticoagulants. That is one. And again, they say it, and I would say it as well. This is not an advice for anyone. Bleeding disorders can be fatal if somebody takes too much of the blood thinners, and that causes, let's say, bleeding in the brain. They can have a stroke and they can even die or become disabled forever. So this is not something that one just start taking these medicines. Some of these, for example, aspirin, are over the counter, but please make sure that you don't take them. Talk with the doctor. Show them this work. I am sure that they would understand what the work says and then provide help. And then they have an additional process, and that is HELP, or plasma apheresis. I personally am not a huge fan of apheresis because of the risk of bringing the plasma out, cleaning it, and putting it back. But if somebody is in dire state, they themselves are in such a bad situation that life has already become very difficult, they are at the edge of disability, then there may be a risk benefit analysis. So here, heparin induced extracorporeal. So heparin induced, let's say they take the blood out of me and then they use heparin to kind of thin it. Extracorporeal, outside of the body. Lipoprotein fibrinogen precipitation. So they take the fibrinogen, these are the clotting factors, and they remove them. So that is the one of the possibilities that they think can be done. So I have given you the solution. So if you just wanted to see the summary, take a snapshot of the screen, this is it. Now let's continue. Now I'm going to go into a little more about the high level pathology. I'll do a second lecture today. We'll not do a chit chat. We'll go over in the second part deeper into their work. So if you would like to hang around and just see the fascinating pictures they have, uh, I would love to. Otherwise, we'll then meet tomorrow. So here, their work. Long COVID, they say, is about 100 million people at this time worldwide suffering from it. However, we all know that diagnosis, there are so many long COVID people who are actually not diagnosed. They're simply told that, hey, this is psychological, or they're told that this is just all incorrect, or you'll be okay, or you just, this happens with time and so on. So the because of misdiagnosis or lack of diagnosis or lack of diagnostic techniques, there may actually be more people than they are diagnosed. So the normal symptoms, again, we all know we've been talking about long COVID for a long time, but just to wrap up or put those symptoms in one place, to wrap the symptoms up, not to wrap up the talk. Fatigue, brain fog, muscle weakness, shortness of breath, sleep issues, anxiety, depression, disability. These are all various intensities and parts of long COVID. Now, here, the long COVID not only damages the lungs, or the COVID not only damages the lung, the pathology is not just in the lung. Even when the pathology is vascular, it doesn't just destroy or cause disease in the lungs. That vascular pathology can be throughout the body. And in that process, the blood vessels themselves can become um, damaged, and the clotting can occur within the blood vessels. The RBCs can become damaged and clotted and aggregated. Platelet can become aggregated. All of these clumps can then become stuck onto the blood vessel. The blood vessel wall can become damaged. That would cause the nearby tissue to have extravasation of the blood and the fluid and congestion and damage, and blood supply will be bad, and the whole tissue can suffer. So here is something that is important from their talk, from their paper. There's tons of material there. I cannot go over all of that in 10-15 minutes. So I'm just going to show at high level what they're talking about. What they say is that when clotting occurs in a healthy person, the clots are really the platelets that are tied together with fibrin strands. So imagine fibrins are like threads and platelets are like little bricks. So you take those bricks and you tie them with threads and you plug them into the injured area and you stop bleeding. That is a normal platelet aggregation and platelet plug formation for normal bleeding issues. Down here, this little picture, this is an activated platelet. So when we have a platelet, remember that platelet is actually a tiny cell. Platelet, it's a small plate. It is actually a pinched off part of a bigger cell that lives in the bone marrow. That bigger cell is called megakaryocyte, big cell, and the platelets are tiny pinches off of that. And platelets, you can say, are filled with glue in them, and so they come out and they circulate in the blood. And whenever there is a breach in the cardiovascular system's closed system, whenever there is an injury, there is an opening in that system, and platelets job is to plug that and close that. So when a platelet finds such an injured area and it comes in contact with certain denuded proteins, it becomes active. When a platelet becomes active, it kind of becomes thin and it becomes spread out. And then more platelets would come in and they would also become spread out, just like band-aids. Imagine these are microscopic band-aids that are stretched over the injured area. And as more and more come and accumulate, then we have these fibrin strands on them as well, and we tie them there. When they had the platelets from healthy individuals in their studies. They formed clotting in artificial chambers that look like blood vessels, and there is blood flow or the water flow, you can say. So they saw that in a healthy person, when the clotting occurs, that clot or the platelet aggregation is on the surface of the blood vessel. And that is how it should be. When an aggregate of the platelets occur, a thrombus becomes a clot appears, it should appear on the surface of the blood vessel to close the breach. And then, secondly, if they have some water pass through that chamber, then the platelet plug breaks down and is washed away. That is a normal, healthy platelet plug's behavior. Good. Now let's see what happens in the COVID patients. And again, they have a lot of information about how this happens. I am skipping that all. Otherwise, we would spend like 2 days over here. I want to show what is the problem and where the solution is. So they say that if they put S1 part or the spike protein in the plasma or in the blood of donors, then the S1 part of the spike protein instigates clotting. And in their article, they mention that this is not that the clotting occurs because the S1 protein causes the ACE2 inflammatory, pro-inflammatory, anti-inflammatory imbalance. I think that happens as well. They're saying that the spike protein itself is a class of its own to promote clotting. It directly acts on the clotting mechanisms and activates them. It does not need the ACE1, ACE2 imbalance. It is causing that as well. So that is a separate imbalance. It directly, when it comes in contact with RBCs and platelets, it can start having them be clotted. So you can imagine the spike protein to be a molecule to cause clotting, not to activate some other molecule which would cause clotting. That's a very interesting observation. So then what happens? I know that there is a part of this lecture that is cut by my picture, but I'll read it out to you. So what happens is, this is the new platelet plug. Imagine that this is a person whose healthy blood is taken, and then S1 proteins are put in it. And what they find is that the resultant clotting that occurs, even when in that plasma there are no platelets. So here in my diagram, I have made the platelets, but imagine another diagram in your head where there are no platelets. Even when there is no platelet, that means it is going to be really difficult to cause clotting. In such plasma, if you put spike protein, the clotting factors become activated. RBCs start clotting, or aggregating, I shouldn't say clotting, aggregating. If there are platelets, all the better, even more clotting. Meaning S1 protein itself does not need a platelet to cause clotting. It would itself instigate other cells and other molecules to start the process of clotting. Scary. So here, when the clot is formed, then the biomarkers that are indicators of the clot formation, for example, plasmins and the alpha antiplasmin and von Willebrand factor and plasminogen and other factors, they all get trapped as well, majority of them. This is the reason that diagnostic labs cannot identify. Then your question will be, well, we do find out that somebody has clotting going on. We find that out by D dimers. D dimer is the second stage after this. When these threads here, the fibrin threads, when they break down, when plasmin acts on them and breaks them down, then the D dimers are formed. So according to these authors, D dimer is an advancer stage. This stage is before the D dimer. And they are saying this stage is not detectable because the markers of this stage are not D dimers. They are plasmins, alpha antiplasmin, von Willebrand factors, and so on. And they are usually not detectable with the normal regular labs because it's stuck here. Okay. The other thing is they saw that when platelet becomes activated, if platelets are present in their studies, they had some blood that didn't have platelet and some had platelet. If platelets were present and S1 was present, the platelet became activated and even damaged, which will actually cause even more clotting because now the platelet itself is damaged and more platelets would try to bind there and more inflammatory system will become activated because there is a local tissue damage, including platelet damage. So membrane damage would occur. Then this part, clotting or the plug formation is not just on the surface, but inside the stream of the fluid. So that means it doesn't really need to be attached to the endothelium. It is really platelets aggregating with each other, platelets aggregating with RBCs, platelets and the spike protein causing other molecules to become aggregating together. And those small microscopic thrombi are just floating around, or emboli, I should call them. If they're not attached to a blood vessel, they should be called embolus. If they're attached to the blood vessel surface, then they're called thrombus. So thrombus and embolus are just floating around now. Then they said those parts that are attached to the surface, it became very difficult to dislodge them by water shower. They were sticky. One, they will not break down. So normal fibrinolysis, normal process of breaking down the clots was not very successful in these clots. And secondly, these clots were stuck to the surface. If they were stuck to the surface, they were in the stream as well. But if they were stuck to the surface, the stream of fluid did not dislodge them. That means they will form fast, they'll form on the surface, and they'll stay stuck to the surface and keep becoming bigger and bigger and cause problem. Now this colorful diagram shows what are the parts of this microthrombus or these tiny clotted things. So the parts here, so just see here for a second, let's orient ourselves. This part is the blood vessel surface. Then in here, if you see, these blue cells are platelets. They are aggregated here. This is their job. They're doing the right thing. These blue strands here are fibrin strands. That is also the right thing. Fibrin should connect with platelets and tie them together. But there are these extra monsters stuck in here. This is also right thing. This scissors like thing, plasmin or plasminogen, this is also a correct thing. Its job is to be stuck in the clot, and then when the tissue plasminogen activator or other plasminogen activator molecules would work, they would activate this, and the scissors job is to break the plug. So our body is clever that whenever we make a thrombus, a clot, our body puts these scissors to dissolve and cut the clot in the clot. So when our tissue has used the clot to block a blood vessel and now it does not need it anymore, and blood not block a blood vessel, meaning block a breach in the blood vessel, and now it is repaired and the clot is not needed anymore, then our tissue releases molecules that activate plasminogen, and that plasminogen would in turn cut down the fibrin strands that would create D dimers, and the clot will be dissolved. So those molecules are here, which are correct. RBCs may be stuck here. There are other molecules that are stuck here. Von Willebrand factor is stuck here. So let me show you what are the things that are stuck. They found that there were red blood cells in it, in the clot. And remember, if the red blood cell is stuck, normally red blood cells do get stuck in the clots. You must sometimes, if you see the clot outside or the little tissue on an injury, you would see it is red in the beginning because there are RBCs trapped in it. But these RBCs have a problem. They are acted on by spike proteins themselves, which further causes the issue with the structure of the RBC, and they start connecting with other RBCs and other platelets. So these are not just good RBCs. Then amyloid protein, these blue little monsters here, amyloid proteins. Amyloid are proteins produced by liver. These circulate in the blood, but in pathological cases, they can start getting deposited in various tissues. And wherever they get deposited, they cause damage to that tissue. They can get deposited on the blood vessels as well and cause tissue there, damage there as well. So they saw amyloid proteins to be stuck there. This is why they called it amyloid clot, because this is an abnormal clot. This is not a healthy clot. This clot has bad things stuck in it because the clot itself was actually not needed. It's an abnormal activation of the system. So it is trapped weird things in it. So amyloid is trapped, which is a dangerous thing. We know fibrin, we know the endothelial cells. Von Willebrand factor. This is also okay to be there, but normally von Willebrand factor, it's a blood clotting factor. It should actually be released out and you could be able to mark it. But if it just gets stuck within the clot, then your labs are not going to pick it up. And they will not be able, before D dimers, you could say from von Willebrand factor, and you could say from the alpha 2 antiplasmin, and you can say from the plasminogens that there is clotting going on. All those markers are kind of less apparent because they're stuck in the clotting, in the clot or the thrombus. So von Willebrand factor is there, plasmin is there, alpha 2 antiplasmin is stuck in there, these green monsters. What is their role? If they are stuck in here, they oppose the function of plasmin. Plasmin's function is to dissolve the clot. These guys, their function is to not let the clot be dissolved. They oppose the dissolution of the clot. The end result is the clot becomes sticky, it becomes resistant to fibrinolysis or resistant to becoming dissolved. And not only that, because of all of these things stuck in it, they would instigate other cells, they would trap other cells, other RBCs, other platelet, and they would just keep adding to the clotting system. Of course, we know that eventually they would cause so much of the clotting that clotting factors and the material necessary for clotting will be eaten up, will be consumed. And now patient is in the thrombocytopenic state. Now they're in the bleeding state. First they were in the clotting state, now they're in the bleeding state, and there is an abnormality there. So if they don't reach the bleeding state and they just keep staying at a lower clotting level, then their smaller blood vessels would continue to become plugged and damaged. And so tissues will get less nutrients. Please remember that the smaller blood vessels like capillaries, their job is to bring nutrition out and take the trash away. If they themselves are plugged, then where would the tissue get the nutrition from? So that is the problem. So once again, I'm done. I said it would be short, still 32 minutes. Summary. Microclotting is an important pathology during acute and long COVID. Vascular damage is not confined only to lungs, but in the rest of, these are the things we knew. I'm just putting them together in one place for anyone who would take just this part and talk with your doctor. Vascular damage is not only in the lungs, but in the rest of the body as well. S1 part of the spike protein is the main instigator of this pathology, and the spike protein does not need to depend on other systems that would initiate clotting. Spike protein itself can start the clotting process. That's horrible. And the clotting type is also not normal. The clot is amyloid clot. They have trapped proteins in them. Labs do not show this inflammatory state. After this state and this clotting state, After this state, when these clots would start breaking down, then the D dimers would start appearing. But according to authors, that is too late. So we have to catch it before the number of clots are too big and the vascular damage is too frequent in the body. And so they're saying that perform the TEG scan. And I'll show you the TEG scan once again. In the next talk, I'll show you how the TEG scan images look and how do they tell us that there is a problem. So if somebody just wants to understand that, hey, what do I do? Then do the TEG scans. And then once the results are in, then doctor has to be very carefully administer antiplatelets and anti-clotting. And they have to be, of course, very careful. They'll have to do tests again and again. They'll have to do bleeding time again and again to make sure that they do not end up causing bleeding in the body. They have to see what drugs are being taken. They have to see what is the immune system status. They have to see what is your tissues status is. Are you prone to bleeding? Do you have any other clotting factor issues? Are there any genetic predisposition towards bleeding? All of that has to be seen. Then this management has to be done. And then additionally, plasma electrophoresis. So I know one part of here is under my name. So it says over here, heparin induced extracorporeal lipoprotein fibrinogen precipitation. That is a way to clean those clots and the fibrinogen material that causes or that helps in clotting. So with this, there is a question here from Aaron. Have they studied what factors cause this to only happen in certain individuals? I do not know if they have done that part or not, but we are seeing that anywhere from 30% to 45% or more are affected, and hospitalized patients, almost more than half are affected. So now exactly what is the reason for them is still need to be looked at. So I'm going to go back to the main page here. And once again, please read this. Number one. Number two, look at this paper. This is also their paper. Thromboelastography is what they recommend. I would just very quickly show it to you. And in the next topic, we would actually go over this in more detail. But if you see here, these lesser ones are the healthy cells, and these are big, sticky, messy amyloid clots. Same here, this is the platelet activation, and these are healthy cells, not much activated. And here are the platelets, and they are activated. So if you see here, fluorescence microscopic example of the different stages of platelet activation and spreading. So that is very interesting as well, and they just keep going. So I'm going to stop here. Please look at this article, share this article. If you don't want to share this video, then share this article. Link is in the description. In here, they have offered various ways to help. So that is the talk. Please do me a favor. Please like, subscribe, and share. This is something that we should share. If you don't want to share the video, then share this article. Whichever way you can tell someone, it can be very helpful. I know that in my own family's cases, these kind of informations were very helpful. So that is one. And secondly, if you would like to support this work, there are links in the description. You can buy me a coffee, or you can be a patron, or you can use PayPal. So thank you very much. I'm going to come back and we would go over the work and the images. I wanted to keep that separate because that can be a deeper dive and take time. Thank you very much. See you soon.